Thanks very much, and good morning, everyone. I welcome you all to this series of seminars. And at the same time, I'm delighted to introduce Amy Crisp. Amy completed her integrated master's degree in chemistry at Lancaster University, and her final dissertation was focused on the optimization of Raman spectroscopy for 3D cell analysis. After that, she joined the engineering department in 2020 as a PhD researcher, and she's working on an interdisciplinary project of translational nature, which is entitled Understanding Biofilms. This project is aiming to improve our understanding of the chemical composition of the biofilm through spectroscopic analysis, where we are using infrared and Raman spectroscopy mostly. So currently, Amy is utilizing spectroscopy to analyze bacterial biofilm samples alongside developing basic microbiology practices. In addition, she is also working on development of anti-biofouling surface coatings by using plasma polymerization techniques to produce coatings which are suitable for application to a range of substrates. The aim is which can resist a biofilm development, and how we can understand them better from their chemical structural point of view by using spectroscopy. So without giving any further details, I will pass on to Amy, who will share her work, her progress on the biofilms she is doing, and she has been supervised by myself and the clinical supervisor, Professor Craig Williams, and then we have in the team Professor Rob Sharp, as well, David Chandler, and also the other team members are Alex Robb, and who is also guiding Amy on plasma polymerization. Amy, over to you.
Thank you, Professor Raymond, for that kind introduction. I'll just share my screen now. Hopefully this is visible. James, could you just let me know if this is visible for everyone?
I can certainly see. Amy, thank you, yes.
Perfect. Okay. So as Professor Raymond said, I'm Amy, a graduate researcher funded by the GISMO project based at Lancaster University, and I'll be giving this talk called Methods to Understand and Prevent Biofilm Progression. So just to give a quick overview of the presentation I'm giving today, I'm going to start by giving some detail on my project and the timeline of work that I've completed to date. Then I'll provide some background information for the work and discuss the significance of antimicrobial resistance in regard to biofilms specifically. Then for the bulk of the presentation, I'm going to go through my experimental work, which is separated into both understanding and preventing biofilms. And throughout these sections, I'm going to highlight the methods that we use to achieve our aims. The overall aim of my work is to improve the understanding of biofilm development, and this is using spectroscopy. Notably, I'm using FTIR and Raman spectroscopy. And in doing this, we're aiming to pinpoint the chemical markers associated with various stages of the biofilm growth. Then, in addition to this work, we're aiming to develop novel thin film plasma polymer coatings with antimicrobial properties, and all of this work is loosely relating to biofilm progression in the wound bed. So I'm now 18 months into this project, and initially I began by training in plasma polymerization. Since the beginning of my work, I've then been producing various polyethylene oxide plasma polymer coatings. When I first came into this project, one of the main aims was to build and develop a new plasma polymer reactor, which is now built and operational in the department. Alongside all of this work, I've also been developing a method to grow and analyze biofilm samples using FTIR. And recently, we've been able to spectroscopically differentiate between planktonic bacterial samples and biofilm samples. So I'm going to start at the very basic level at the beginning, just by asking what is a biofilm. So individual bacteria are known as planktonic, and then you have biofilms, which are bacterial communities that can form on any surface, and they're classed as irreversibly developed once the extracellular matrix is formed. This matrix then allows communication pathways to be established both inter- and intracellularly in a process that we call quorum sensing. The biofilm is actually the favoured state for bacteria to exist in, and once it's irreversibly developed, it can cause quite a lot of challenges for us. Biofilms exist everywhere, so if you run your tongue across your teeth right now, chances are you're going to feel a biofilm. Biofilms are present in so many different areas that have impact on human life, and they can be beneficial or problematic. They notably have an impact in the food industry, in water distribution systems, and in marine enterprises, so on the bottom of boats, causing damage. However, biofilms are cause for significant concern in hospitals and medical environments, both where equipment is concerned, for example with urinary catheters or hip implants, which suffer most commonly, or directly on the patient, so in the wound bed. And many bacterial infections that form biofilms are much harder to treat because they're much more resilient to the treatment options that we have. My work itself is based around biofilm prevention, and as such, a lot of this presentation is going to focus on preventative strategies. So I'm just going to quickly summarise the development process that takes you from a single bacteria round to a biofilm. So it begins with a single bacteria that reversibly adheres to a surface, and this happens when it's close enough. This is just through weak interactions like van der Waals forces, and then more bacteria can start to join. And this is when you get a monolayer forming across your surface, which allows for the production of extracellular polymeric substances, forming this matrix around your bacteria. It's at this point that the microorganisms are now classed as irreversibly adhering to that surface. From here, you get the formation of multi-layers, which then paves the way to those communication pathways, and nutrient channels can start to form. This is then when you would consider it a mature biofilm. Once that's happened, you can actually get other microbes in the near vicinity joining, so it's very rare that a biofilm will be formed from a single strain of bacteria. And at this point, the bacteria can actually start to detach, so enzymes are released to break down that matrix, and then bacteria can begin to colonize the surrounding area. So you can see it's kind of a circular process, and it will just keep on going if you don't do anything to prevent it. Excuse me. So particularly as I've been examining surface modification techniques to prevent this biofilm formation, it's vital to consider how the process starts and what happens to allow that bacterial attachment in the first place. So this process commences with a so-called conditioning layer that forms on your target surface, which is often going to be a solid liquid interface. This layer is formed from organic and inorganic materials that are secreted by the microorganisms in the bulk liquid adjacent to that surface. This is why the layer then differs significantly for each bacterial strain, and it differs under different environmental conditions. It's this layer that's priming your surface for that bacterial attachment. Another important part of this cycle to highlight is the formation of the matrix from these extracellular polymeric substances. Once this is established, there's not any current antimicrobial therapies that can fully eradicate it. The EPS itself is made from a highly hydrated mixture of proteins, polysaccharides, and external DNA. And importantly, the structure is dynamic, so it's known to change composition depending on the microorganisms themselves, but also depending on physiological status, nutrient availability, and external stressors. So as you can see, biofilms are extremely adaptable, and there's lots of factors to consider, which is why they can be proving to be such an issue. Now, the motivation behind my project and much of the research in this area comes from antimicrobial resistance. So AMR is an evolutionary certainty. It's effectively the response of the bacteria which are fighting for their own survival. But antibiotic resistance is now endangering public health. By 2050, it's expected that the number of deaths from AMR-related issues is going to rise to 10 million per annum. And this is more than the current death toll from cancer worldwide. Bacterial infection is increasingly resistant to available treatment options, and the bacteria within a biofilm are estimated to be 1,000 times more likely to resist that treatment compared to the planktonic bacteria. There are several factors which are accelerating this issue. If you just consider planktonic bacteria alone, you have the increased use of antibiotics, within which you have the selective pressure of microbial use, but you also have susceptible hosts, mistakes in infection control programs, microbial characteristics, but biofilms themselves are a separate and additional factor that are increasing antimicrobial resistance, and this is obviously the most important for us to consider. Biofilms have unique properties that allow the resistance of antibiotic treatment. They can even resist the attack from the human immune system. So this just adds an additional layer of complexity to the issue of AMR. Firstly, if we just look at the biofilm's natural defense mechanisms, it has the EPS matrix. As I mentioned in the previous slide, this is formed around your bacteria, and this acts as a protective barrier to the surrounding environment. It's actually been shown to prevent dehydration of the biofilm, and it doesn't always permit antibiotics to penetrate within the main biofilm. But there are obviously cases where antibiotics can penetrate into a biofilm. In these instances, more issues just arise. So often biofilms are established so that you have the core cells, which are virtually inactive or physiologically dormant, and they're surrounded by the working cells. This means that if the antibiotics do penetrate and they're killing off those outer bacteria, they're not able to harm the inner bacteria because antibiotics are relying on a cellular activity to produce their response. Then, even in these cases where you have effectively destroyed some of the biofilm, that mass will just remain on the surface to be consumed by any surviving bacteria. So the whole process can just continue anyway. What we need is to completely disrupt and prevent the regrowth of this extracellular matrix, and ideally you want to prevent the formation of the matrix altogether. Now, aside from these natural defense mechanisms biofilms have, resistance can actually be acquired through genes between microorganisms. And this is something that happens in biofilms more readily. So due to the communication mechanisms that are in place in biofilms, AMR genes are exchanged between bacteria more readily by horizontal gene transfer. And actually, interestingly, it's been demonstrated that reducing nutrient availability within biofilms doesn't actually diminish this ability to participate in HGT. So as you can see, biofilms are really quite tricky to deal with, and antibiotic resistance is just making this problem even worse. So now I'm going to move on to the research that we've been conducting here at Lancaster, and I think a nice place to start is on the understanding side of biofilm research. We've been conducting vibrational spectroscopy to study Staphylococcus epidermidis biofilms. Firstly, I'm going to run through the specific methods we use and explain a little bit about the principles of spectroscopy for anyone who's not familiar. Spectroscopy is the study of the interaction of electromagnetic radiation and matter, and this is as a function of wavelength or frequency. By conducting this analysis of a sample, we produce a plot or a spectrum which will demonstrate chemical information about your sample. Our work often focuses on vibrational spectroscopy, and this just elucidates properties about your sample based on the electrons in your sample undergoing some sort of vibrational transition. In our case, we're employing both Fourier transform infrared spectroscopy and Raman spectroscopy. Light itself can be viewed in two ways, so you have particle theory or wave theory, and in the case of our spectroscopies, it's really useful to think about photons or individual light particles coming into contact with your material. So I'm sure everyone is aware light can interact with molecules in various ways. It can either pass straight through in transmission or it can be reflected. Under particular conditions, photons can be absorbed into your material, or alternatively, the photons can be scattered. And these different processes are utilised in different spectroscopies. So in our case, for FTIR, this focuses on absorption and transmission. And then you have Raman spectroscopy, which implements scattered light. So I'm going to explain Raman spectroscopy first. The majority of light that's scattered is going to have the same energy as the instant light. So this is referred to as Rayleigh scattering, and it's an elastic process, which means it occurs without a detectable change in energy. The much weaker counterpart of scattered light is then Raman scattering, and this results from the instant radiation interacting with your molecule to induce some sort of nuclear motion. And what this means is that the photons have the potential to be scattered at a different frequency to the one that they came in at. This is now an inelastic process. Raman scattering doesn't commonly happen. So realistically, only one photon in approximately every 1 million photons is going to be Raman scattered. But Raman spectroscopy exploits this scattering to yield chemical information about the molecule you're analysing. The sample analysis process works with a monochromatic light source that's directed towards your sample, and then this radiation is transmitted or scattered. The resultant spectra are then a plot of the intensity of this scattered radiation as a function of the shift in wave number from the instant radiation. Oh, sorry. So Raman spectroscopy itself is benefited by chemical specificity, and it's usually a label-free process, it's non-destructive, and it's generally known for producing unique spectra for every given substance that are described as fingerprints. This is because the peak shapes and intensities relate so specifically to the molecular structure in your sample. Specifically, I use Raman microspectroscopy, and this is the combination of standard Raman with a confocal microscope, which allows for single cell analysis. So this technique is slowly becoming more established for microbial cell analysis, and we actually have a spectrometer available and in use at the University of Lancaster. So here, if you see in this picture, you have the sampling area here where you place your, often a slide, or it can be anything really. It's placed under this microscope, and then the laser is processed in this section here in various grating systems, and it's directed towards your sample. This whole process can then neatly be controlled by the computer adjacent to the system. Another vibrational spectroscopy that we employ in this biofilm research is Fourier transform infrared spectroscopy. So Fourier transform is just the name of the mathematical method used to convert the spectra to a more easily read format, and this is done computationally. Infrared spectroscopy itself is the use of the IR component of the electromagnetic spectrum. So you expose your sample to this IR light in your spectrometer, then some photons are going to be absorbed by the sample and some will be transmitted. The spectra then result from measuring this absorption or transmission, which is going to be unique for each chemical bond. So again, you can produce this detailed information about the chemical composition and functional groups you have in your sample. We conduct this work with the help of the RLI pathology labs, where we have this desktop FTIR spectrometer, which is quite a nice, neat piece of kit. So your sample goes in this in-transmission sampling area. We do have a ATR setup as well. The laser then will pass through your sample through here, and you'll get your spectra reading on this little computer here. So in our biofilm study, currently we're primarily utilising FTIR, and at the moment we're aiming to use this spectroscopy to differentiate between the chemical composition of the biofilm at the various stages of growth and determine whether the EPS is homogeneous. Following this, we'll then progress to Raman spectroscopy, and this will allow the measurements to be taken for live biofilm samples. So Raman allows you to monitor samples without harming the bacteria. In doing this, we'll then be able to progress to showing how the substrate affects the growth of the biofilm and see if we can pick this up on the spectra. Also, a nice idea would be to compare different types of bacterial biofilms and determine whether they can be differentiated with principal component analysis, which would then have applications for clinical diagnosis. Quickly, just to demonstrate my biofilm growth method, just for reference, we use a fairly simple glass growth chamber, and a nutrient broth is then maintained at a particular concentration. We put calcium fluoride slides into this broth and then incubate it for the desired time periods. For our FTIR work that we've completed to date, samples are then removed at your incubation period and transferred to a drying chamber just to partially dry the samples. So in the early stage of testing, this just allows the removal of the bulk of hydration, which can overwhelm FTIR spectra sometimes, which can then lead to masking vital signals. So as an example, if I show you these two overlaid spectra, we have a 24-hour aged biofilm compared to the spectrum of a planktonic bacteria. So each peak in this spectrum is then tabulated, and there are then standard tables of these peak assignments from which I can determine this information. Each peak relates to a specific chemical signature within your sample. So, for instance, if I take this amide 1 protein group, typically this will sit between 1600 and 1700 wave numbers. The exact peak position is then controlled by the conformation and type of protein bonds that you have. So from tabulating all of our data like this, we can infer two key things. Firstly, you have a change in peak position, or the wave number, and this relates to slightly differing chemical structures that cause that signal. Then you also have increasing and decreasing absorptions. So if you have, for instance, this peak at the same wave number, the increased absorption indicates that you have a different abundance of that same chemical component. Through the work that we've done so far, we have spectroscopically differentiated between biofilm samples and planktonic samples that have been formed from the colony used to make that biofilm. So I'd like to draw your attention to 4 of our key findings. Firstly, if we look at the higher end of the spectrum, we have the lipid region. So this reveals water content, which is obviously much broader for the biofilms. The biofilm is this top peak here, and then you step down to the planktonic sample. Now, I did mention that we're using partially dry samples, but the biofilm still has an intact extracellular matrix. So this is going to contribute moisture that the planktonic cells don't have. Now, the most important differences are actually seen in the lower half of this spectrum. So firstly, I'll draw your attention to the amide 1 peak. This definitively shifts, so you have different proteins existing in your biofilm compared to your planktonic sample. And if you think about it, this is quite logical, because with the biofilm you have that extracellular matrix, which we already know contributes these new proteins. Then again, if you move down the spectrum, you see this peak at 1082 wave numbers. So here you see a shift in the peak, but also a change in the shape. And again, this is just alluding to fresh proteins in our sample. Finally, we have a very interesting occurrence in this DNA backbone peak, where 2 key peaks in the planktonic sample seem to coalesce in the biofilm sample. And what this is actually indicating is that there's a fundamental change occurring within the bacteria of the biofilm. And at the moment, this is where we've got to with this work. So far this is indicating that biofilms can be distinguished in this way, and it's actually suggesting a future possibility for rapid clinical sample analysis. So now I'm going to move on to the other side of my biofilm research, where we've been evaluating plasma polymer coatings as a mode to prevent surface biofouling. Plasma polymerization itself is a surface coating technique established in the 60s, and the process works with a monomer that's activated with radio frequency power to produce a plasma discharge. This activated monomer can then partake in various fragmentation and rearrangement reactions, which initiates polymerization. By simply placing a substrate in the reactor during that process, the fragments are going to naturally combine at that surface, forming a cross-link structure, which is then your plasma polymer coating. These coatings are both uniform and stable, and in doing this process we actually have the ability to produce ultra-thin film coatings. So for example, I work with samples that are between 1 and 100 nanometers in thickness. Plasma polymerization is also benefited by the ability to control plasma parameters, and this allows you to tune the surface of your material for various applications. So in our work, the coatings are obviously geared towards anti-biofouling. And I would like to actually just state here that any material that you have can technically be used as a substrate to plasma polymerize onto, but in our case we use clean silicon wafers, and this is just selected for ease of the sample analysis. Just to summarize that process using this image here, this is the existing reactor that we have at Lancaster, and the process works with this reactor glass barrel, which you maintain at approximately 0.5 Pascal. The chamber is then filled with monomer by opening this monomer inlet valve here. So then monomer moves into your chamber, increasing the pressure. So pressure is the first plasma parameter that we have to control the surface. The next vital component for the process is then to have an electric field. So to achieve this, we employ an RF generator, and in this case a matching network. And this is connected to a copper coil around your reactor. When you turn this power on, the monomer is then activated, producing this plasma, which you can see in the glow. So power is then the second plasma parameter that we have to control the surface. At this point, you then have a plasma in your reactor, and as I said in the previous slide, at this point you have different fragmentation rearrangement reactions going on, and you're able to produce your coating. So to do this, you would place your sample here so that it sat in the plasma glow. The third and final plasma parameter that we have is the deposition time, or the time that you allow your substrate to sit in this plasma. And this is used to control the thickness of your coating. So just as an example, an ethanol plasma polymer coating that I've produced at 10 pascals with 30 watts of power and approximately 15-minute deposition time gives a coating of around 80 nanometers. Oh, I should also briefly state that all of the plasma work that we do tends to use continuous wave plasma, but you can have the option to use pulsed plasma using this oscillator in this picture here. Now, I mentioned at the beginning of this presentation in my project timeline that one of the key objectives coming into my project was to design and build a new plasma reactor. So here is the initial drawing and pictures of the reactor in situ in the physics department here at Lancaster. So if you just note this bottom image to show the reactor in use, this is actually not with any monomer. So this is just opening a valve to let air in, so it's an air plasma, and this coloration comes from the nitrogen in the air. Due to the nature of plasma polymer reactors, the same parameters are never going to yield exactly the same coatings from reactor to reactor. So there's always going to be slight variations, and that's just a fault within the field. But when designing our new reactor, we did take every effort to try and maintain the necessary and important features of the existing reactor that I showed here. This just means that coatings are going to be comparable to those produced in the original reactor. One feature to note, obviously, in this new reactor is the use of a cruciform reactor setup. And this just allows the use of multiple monomers, so you could have multiple monomer surfaces, but it also allows us to combine analysis with production, so you could monitor the plasma as it's depositing on your substrate. Now, although this cruciform barrel is currently in place, all of the parts that we have, like I say, are adaptable, so we can actually put a straight barrel in this setup. And this reactor is currently used in work that we do with businesses on the GISMO project. Now, I have mentioned that varying the plasma parameters is going to influence the coating properties, but before I expand on this, I'm just briefly going to explain the primary analytical technique that we use to evaluate these coatings, and this is X-ray photoelectron spectroscopy. And we're lucky enough to have an XPS spectrometer available in the physics department at Lancaster. XPS is a surface technique, so it's sensitive to the top 5 to 10 nanometers of your material, and it's used to quantify the elements on that surface and evaluate chemical state information. You can use X-rays of a known energy and direct them towards your surface. Then electrons will be ejected, and you can calculate the binding energy of these electrons. So the binding energy of the electrons depends on the core level from which they're excited. Hence, you can sort of work backwards and determine the elemental composition and also the chemical environments for each element. In this project, the initial test work we did was carried out with ethanol, just as a simple starting monomer, and these results here neatly demonstrate how altering those plasma parameters is going to influence the surface of your coating. So we produced ethanol coatings using different pressures and powers, and then analysed those with XPS. Now, initially, a wide scan spectrum is going to be really useful to demonstrate the elements. So this is the spectrum that I showed on this previous slide. The so-called high-resolution spectra for each element. So these are both C1s peaks. Then within these, you have peak analysis showing the different bonds. So if we first look at this overlaid spectra, you can see the differences that are occurring because you've altered those plasma parameters, so you can see these peaks shifting about. If we look at this individual spectra here, you have 3 key carbon bonds that we want to keep an eye on. So you have the hydrocarbon peak that sits around 285 electron volts. You then have the CO bond at approximately 286 electron volts, and then the C double bond O at 287.5 electron volts. And you can see here that as you're shifting those plasma parameters, those bond peaks are shifting. And often what you're aiming to do is maximize the character of one of those bonds, which I'll come on to soon. What we discovered from this preliminary work with ethanol was that a combination of high pressure and low power maximized the non-fouling characteristics of our surfaces. The main focus of my work in this area over the last 18 months has been attempting to incorporate polyethylene oxide to achieve these non-fouling surfaces. So ethylene oxide units are already known to reduce biomolecule absorptions. And as I've already mentioned, ethanol was a really good starting point for this work, but from reviewing the literature, a really viable option seems to be crown ethers. So in the early 2000s, 12 crown 4 was explored by the Timmins group as an option to produce these non-fouling coatings. And these are the XPS spectra they produce. So you can see they have one peak which is clearly dominating the spectrum, and this is the peak that sits at around 286.5 electron volts, which relates to the ethylene oxide character, or this CO bond. This is what we're aiming for in our research. Now, in addition to these crown ethers, we work with a company that have developed an ultra-thin film coating, and for this instance, we're going to call them our industrial standard. So as you can see in this XPS spectrum, they've really maximised this ethylene oxide character, making it an ideal anti-biofouling coating. Now, despite the smaller 12 crown 4 monomer being used in the research that we've based our experiments on, we decided initially to work with 15 crown 5. This is just because it's much safer to work with and it incorporates more carbon and oxygen, so theoretically you would assume it would make better coatings. Initially, we began with deposition methods similar to those we used with ethanol, so we were using a high pressure, low power regime. But what we found was this monomer is very temperamental. It's quite viscous, so it's very challenging to actually vaporize in the first instance. And even in the coatings that we did produce, we're not maximizing the ethylene oxide character in the way we'd want to. So this hydrocarbon peak is clearly dominating the spectra. So obviously the next step is to move on to using 12 crown 4 as the monomer. Now, we haven't actually implemented that at Lancaster yet, but our colleagues that we work with in Liverpool University have already begun preliminary studies with this monomer. So as it stands, they've tried various plasma parameters and they have successfully produced coatings. And as you can see in this C1s XPS spectrum here, they have maximised this CO content. And it's obviously much higher than what we saw in the 15 crown 5 coatings. So hopefully over the next 18 months, what we expect is to see these coatings just improve further and eventually begin to approach those levels seen in the industrial standard. Alongside our production of coatings, we've been looking at methods to evaluate anti-biofouling coatings. So when I say anti-biofouling, I mean surfaces that are able to resist proteins, bacteria, and virus. So before you consider directly exposing coatings to bacteria, it's a good idea to look at some of the key proteins employed by cells and bacteria to determine whether the surfaces are even viable as non-fouling. So we selected albumin and fibrinogen because they're already known to be vital for bacterial surface attachment. These two proteins are actually observed in that conditioning layer that's priming surfaces for bacterial attachment that we saw right at the beginning of the biofilm development cycle. So in our experiments, we exposed our ethanol plasma polymer coating and the industrial coating to each protein individually for 1 hour. Then we used XPS spectroscopy to monitor the nitrogen content on our surfaces. So prior to our protein tests, our samples don't actually contain any nitrogen. Hence, you can sort of assume that any nitrogen afterwards is relating to that protein adsorbing to the surface because proteins obviously contain nitrogen in their structure. This table here then displays the nitrogen counts, and these have been normalized to a gold standard within the spectrometer. So XPS, the counts will vary naturally day to day, and we're obviously collecting data over various months that we need to be able to compare. So from this data, what you can see is that the industrial standard is obviously much better at resisting the proteins, which we would obviously expect. But interestingly, what it also shows is that the fibrinogen more readily binds to the surface compared to the larger albumin molecules. So what are the next steps in our experimental research? Well, in terms of understanding biofilms, we're going to start looking at examining the biofilm growth from 1 hour post-incubation up to 96 hours. And what we're trying to do is define this point of irreversible attachment to the surface, so that point at which the matrix is forming. Then we're also going to be moving away from FTIR and onto Raman spectroscopy. And this is going to allow live cell imaging. So with Raman spectroscopy, you can actually have your samples incubating and collect spectra at the same time, so you can monitor the growth in real time. Then for developing preventative strategies for biofilm progression, we're going to move on from our individual proteins and look at applying blood plasma and monitoring that attachment compared to those individual proteins. And this will soon progress on to exposing our surfaces to live bacteria. So that will be a crossover point with the bacterial culture work that I've been developing as part of the understanding side of this project. Alongside this, we'll also be producing our plasma polymer coatings from the 12-gram-4 thermometer, and this is going to be hopefully getting them optimized for that ethylene oxide retention. Just to summarise, here at Lancaster, we're currently simultaneously working towards improving our chemical understanding of biofilms by employing that spectroscopy to monitor the chemical changes in bacteria. But we're also, in parallel to that, evaluating plasma polymer coatings as a scalable and viable option to prevent biofilm formation. So I'd just like to quickly thank my supervisors again for their support during this project, as well as the lab groups that I'm a part of. I'd also like to acknowledge our collaborators at both Liverpool University and Glasgow University, as well as the Royal Lancaster Infirmary Pathology Labs, where I conduct a lot of my bacterial work. And I'd also just quickly like to reiterate that all of the equipment that I've mentioned today is currently used and available through the GISMO project. So thank you for listening. I'm happy to take any questions anyone might have.
Thanks for that, Amy. And yes, if you'd like to ask Amy a question now, you can either put it in the chat, or if you're feeling particularly brave, you can unmute your microphone and ask it directly. I'll start with one, Amy, if that's okay. As you know, GISMO is a project that's aimed particularly at supporting businesses with research and development. And I wonder if you could just perhaps summarise the implications for businesses with an interest in anti-biofouling products and qualities in terms of things like new product development. So what have you found out from your research? What are the implications, if you like, that for product development?
Okay, well, I think perhaps the best way to go at that question would be to use examples. So I'll obviously be careful about what I discuss. But one of the things that we have been doing is using XPS to analyse samples companies have sent to us. And by doing this, you can tell them exactly what's on their surface, and then they can work out what additives to use. So you can tell them what bonds are available on that surface to react with. And then that would obviously have implications when you're considering producing anti-biofouling surfaces that don't use plasma polymerization.
Okay, thank you. We've got some questions coming through, and Itisham, I can see you've raised your hand as well. So, Itisham, did you want to say something?
Yes, Amy, excellent presentation, and you're making an excellent progress. Just a very quick question. You're using spectroscopy to look at different bacteria. So, which bacteria you have analyzed so far, and what you are planning for future? What will be the other bacteria you'll be analyzing?
Yes, so currently we're using 2 different strains of Staphylococcus epidermidis. We'd also like to look at MRSA, obviously that's very clinically relevant. We'd also probably like to expand the study into looking at different candidas. So they're actually used by our collaborators in Glasgow, so that would be quite useful to have.
Thank you.
Okay, we have 2 or 3 questions coming up in the chat. Rob Short asks, could you comment on the scalability of plasma? For example, does it support existing products?
I don't have specific knowledge on any, but I do believe there are some that exist. It's something that you can quite easily scale up, but like I said, there are issues with the actual functionality of coatings between reactors. There are always going to be slight variations, but as far as I know, they do exist currently.
Okay, thank you. And some comment from Terry Swanson, who says, thank you for your excellent presentation. And he says, you talk about antimicrobial resistance. Is it true resistance versus tolerance?
Oh, I've not come across that. What does resistance versus tolerance mean? Sorry.
Terry, I think you're on the call. Would you like to ask that one directly?
Yes, hi. I'm not a scientist, I'm a nurse. And we talk a lot about with biofilm that they are tolerant to the antiseptics and the antimicrobials we use versus resistance. So this is more of a clinical versus a science. And so I think sometimes the terminology of resistance and tolerance is used interchangeably with but it has different connotations, I would say, in different scopes of practice.
Okay, so I would suppose that goes back to one of my earlier slides where I was discussing how the biofilms naturally resist treatment. So maybe that is more, they're tolerant naturally to treatment, but they can also acquire that antibiotic resistance. Hopefully that answered your question.
Okay, couple more questions coming through, Amy. Harsha asks, will you be exploring mixed species biofilm models?
Yes, so that is something that we're really interested in. Obviously, it's very rare, like I say, to have single strain bacterial biofilms. So that is something we'd like to do. I'm quite limited on time in my project, so I only have 18 months left. But if the availability happens, we would like to do that, yes.
Okay, all right, thank you. And Craig, you had your hand up and you put it down again very quickly. Just wanted to check to see if I'd like to ask a question.
No, it was just really a comment, being as kind of Amy was struggling with the tolerance resistance thing. But I think Terry kind of hits upon a really interesting point, because in biofilm modeling, most people have used kind of type organisms rather than clinically resistant organisms. So we don't actually really know what would happen if a resistant organism becomes incorporated into biofilm, because you then expect it to be a kind of like a combination response of the inherent mechanism of resistance within the bacteria, plus also the physical differences of getting the antibiotic to the biofilm. So I think that the tolerance and the resistance is more kind of mechanistic rather than in terms of clinical outcome, but it's an area we'd hope to have a look at when we start to look at more resistant organisms in the biofilm model.
Okay, thanks for that. Well, I can see perhaps one more question or comment coming through the chat. Somebody's busy typing away. Two more questions. Good grief, Amy, you're going to beat the record for questions asked, which is going to be good. Well, your supervisor's saying it's an excellent and very clearly presented, so that's got to be a good thing, hasn't it?
Very kind.
Yes, if we'll probably make this one coming up the last of the comments and questions before we move on. But it's like some commentary here from Harsha. There are guidelines currently being drafted by ECHA related to the definition of tolerance and resistance in terms of biocides and biofilms. Watch this space. Good grief. Excellent. Okay, well, thank you very much indeed for those questions. And Amy, thank you for responding to them and for your excellent presentation just now. What I'd like to do now is perhaps just very quickly speak to you all about the GISMO project and the help that the Lancaster University can provide generally. GISMO, as I mentioned before, is a project that's being delivered by Lancaster with support from the European Regional Development Fund, and that's all about helping businesses to improve the way that they design and use materials. And whilst the GISMO project is specifically for businesses in the Cheshire and Warrington area in England, the kind of support that the university can provide is the same across the board, really. And what we've tried to do on the GISMO project is to capture that support by way of what we've called, in this particular case, a materials MOT. So people from the UK will be familiar with putting their car through the MOT to make sure that it's the tests and checks to make sure that it's fit for purpose and fit for the road. And at Lancaster, we offer to do that for your materials too. So picking up on the theme of today's presentation, We can help you if you're interested in antiviral and antimicrobial resistance and protection and effectiveness. And we can look at the characteristics and effectiveness of coatings and look at the material characteristics generally to check that they can support the work that you'd like to do or how you may need to improve them. But we can also help you with the availability of new and alternative materials and methods. If you're particularly driven by environmental sustainability, and who isn't nowadays, we can help you to look at how to achieve that. If you are creating new designs and formulations and you want to be able to check their accuracy and effectiveness, if it means, for example, creating prototypes for you, particularly through things like 3D printing and 3D modeling, we can help with that. Or purely if you want to just benchmark the performance and characteristics of your materials with those of others, or your products with those of others, we can help with that. And particularly, of course, if you're looking to incorporate smart materials or properties into your products, that's very much what our researchers and Lancaster's GISMO project is all about. So if any of those are of interest to you, and if you want to engage further with Amy and Nitisham and the team at Lancaster after today's presentation, we'd be delighted to do that. And just to let you know also that we've got another seminar up and coming in about a month's time, when one of Amy's colleagues, Noah Wright, will be looking at frustrated Lewis pairs, hydrogenations and their applications in flow chemistry. Well, I'm going to turn up to that just to make sure that I understand what all of those words mean. But if that's an area for you, you can book onto the event using the link that you can see on the screen there, and we'll be circulating the details of that to you after this event, together with a link to the recording of today's seminar. So if you want to be able to go over things again, we should be able to make that recording available to you in the next 24 hours or so. And if you do want to get in touch with GISMO, or if you want to get in touch with the colleagues at Lancaster, you can do so through the GISMO project initially. Whether you want to check out our website or follow us on social media, we'd be delighted if you're able to do that. Or if you just want to email to get in touch, there's the email, gizmo@lancaster.ac.uk. And the telephone number that if you rang that now, my phone would buzz next to me. We'd be really delighted to hear from you. But that having been said and done, I'd like to thank you for attending this morning and for your questions and the insights, particularly from Amy and Nitisham. And that's it then. Thanks very much again. Hope you have the rest of a good day and good afternoon.
Thanks very much, James, and thanks very much for attending. Well done, Amy.